Just full screen it.
Okay. So then the note section wouldn't be
Yes,
visible
I
as.
got your point. Got your point. So
Yes,
that
sir.
yes, so Astha took your this thing prepared was by Astha. She is here. So her question is somewhere it is written in millimolar and somewhere in micromolar. So which one is right?
Yeah, so whatever I've written in the notes section, that is the correct concentration. Actually, while speaking, I had spoken it wrong.
Okay, ma'am. Okay, ma'am. Thank you.
Yeah, so it's millimolar.
And ma'am, I had another doubt in slide 9 here. In this section of suggested PCR program, I was not able to understand what does it exactly mean. I mean, because segment 1, 2, 3, and then this data I was not able to comprehend properly. Should I do it in the slide? I mean,
Yeah, please,
slideshow.
please.
Yeah.
So this is the PCR cycle that you're supposed to input into the machine. So the 95 degree is the initial denaturation that we do. So that would be done for 2 minutes. So
Okay,
that
ma'am.
is the number of cycles is one. So that is just one step that you have to do initially.
Okay, ma'am.
So
And okay.
the
Uh,
second
so
segment, the second segment is the one that has to be repeated for 30 times. So that is it has three separate steps. So that is denaturation, extension, denaturation, annealing, and extension. So the duration is mentioned respectively. So first we have 95 degrees denaturation, which would be done for 30 seconds. Then the primer TM. So as we calculate the annealing temperature, this is what it is given, that usually the annealing temperature is taken below 5 degrees or around 5 degrees of the melting temperature of the primers. So that is done for 30 seconds, and the extension is done for 72 degrees for 1 minute if you have the length of the target is less than 1 kb, or if it's more than 1 kb, then you need to keep it at 1 minute per kb.
okay, ma'am.
And the third segment is the final extension that we do. That is also repeated just once. So the number of cycle is given as 1, and it is done for 10 minutes at the end of the entire program.
Okay, ma'am. Okay, ma'am. Understood. Thank you. Um, just a second.
Anyone else?
Yeah, I have two doubts. One is about making the shRNA. I just have some doubt in what the final thing, the setup that we are making. Uh, how is that ultimately going to give us the final shRNA, as it was given in the slides? And the second thing is, whenever we are making a fusion construct, it is said that the first out of the two proteins, the first one should have the start codon, and it should not have the stop codon. In some cases, even in the second uh, protein, we have the start codon included. What difference would it make if whether it's there or not? And another thing is, uh, in the NCS, we are cloning it, like we can choose any two restriction enzyme sites. How do we choose them in such a way that our proteins ultimately come in frame to the, like, should the distance from the promoter or the RBS need to be taken into account while choosing restriction sites?
Maybe we can go one by one your questions. So the
Okay, sure, sure.
fusion thing will go first. I think Rahul taught it, but let me try to explain it. If I am wrong, then he can correct me.
Okay.
Okay. So your question is whether if your second protein has a start codon, so do you need to omit it or you can let it be, right?
Yeah.
So I don't think it should be a problem if your second protein has a start codon, because the ribosome binding site is where the Kozak sequence is in case of eukaryotes, right? So it is in front of your first protein, right? So the Kozak sequence or in case of bacteria, it is called Shine-Dalgarno sequence. The ribosome will bind there, and from there the translation will start. So even if there is another start codon, in some cases ATG usually codes for methionine, right? many proteins have ATG in there first, and even in the middle, do you think there is no methionine? There
Yeah, there
obviously
is.
will be also
Mm-hmm.
methionines, right? ATGs will also be there in between sequences after the start codon. So what matters is the Kozak sequence or Shine-Dalgarno sequence that will initiate your translation. So if you can omit your second, I mean, even if your second protein has a start codon, it will not be a problem until and unless there are sequences like Kozak sequences present. Like after ATG, I think the fourth position usually is a G or something. I don't remember it correctly. What is the definition of Kozak sequence? So I don't think it should be a problem. And
Okay.
regarding your first question, like how shRNA is generated, how does it function? So shRNA and siRNA both have like similar kind of mechanisms, but in shRNA you have to clone it and then it gets processed. Pretty much is covered in the slides if you just look at it.
I just had this doubt that in the final construct they were using like when you explained it we are using two restriction enzymes to clone it.
Mhm.
I could not understand those particularly. Like where are they? Like why have we put them the where they have put where it has been put?
Okay, I don't think I'm following your question. I think there is can you do one thing?
I'll
In the
open the
right side.
slide.
Yeah, yeah. Or maybe you just do one thing. See, in the doc, we have shared a doc file. Okay,
Mm-hmm.
just put your question there. I'll get back to you. I'll
Okay, all
email.
right.
Okay.
Okay, okay,
So the point is, if we can't answer any of your questions, you just put it in the doc file. I will get back to you. And what we
okay,
can answer right now, we'll try our best to answer.
okay. One more thing was, so in the case where we had to in the assignment we needed the his tag, so we used the vector which already had a his tag with it. So in
Uh-huh.
that case, our protein and the his tag were not like there was a
So in
little.
frame wala
No,
confusion.
no, they were in frame,
Mm-hmm.
but so they are in frame, but they have nucleotides in between them. Maybe six
Right,
or
right.
nine were there. Versus if we would have cloned them the way we did it for the flag tag, there would not have been any nucleotides in between them.
Mm-hmm.
So from the perspective of like the way these whenever we are making a construct, if we have a choice of following either of the two ways,
Right.
what difference would that make, if at all any?
Okay, okay. So, right, I think I got your point. But if it is a small stretch of peptide that is not disturbing the codon sequence,
Mm-hmm.
okay, then it should not be a problem. But
Okay.
ideally, obviously, you will want it immediately starts after your protein, right? I think that
Mm-hmm.
is better. But if
Okay.
there is a blank peptide sequence which does not alter the frame, I mean, no frame shift mutation is happening, then it should be okay.
Okay, all right. Yeah, because like in case if we are cloning them right one after the other,
Right,
we are
right.
very sure that
I
they
got
will
your
follow
point.
the
In some
frame.
cases, actually, it is desired also. Key, you give some
Uh-huh.
difference of basis. For example, if you're working with RNA, in that
Mm-hmm.
case, it is desired you put some flanking sequences so that the transcripts are formed well and it is processed by the IST complex better in a better
Okay.
way. So
Okay.
immediately. But in case of protein, it should not be a problem. But that one thing you have to keep in mind that the shift doesn't happen, right? So
Okay.
other than that, it's fine.
All right. Thank you so much.
Okay. Anybody else? Even if any of you are not having questions right now, but you can share it in the doc file, we'll get back to you.
One more question.
Yes,
So,
Pallavi.
when I was working on the assignment and we had to make the siRNA, so I used SI Direct tool.
Okay.
So I got only two candidate siRNAs.
Right, right.
Now, both of them had significant off target effects, as in, out of the 19 nucleotides, 16 were matching in one of the cases.
Mm-hmm.
What can I do in such a situation?
Okay. Then you should have tried another tool, but it's fine. I think you guys have submitted the assignment, right?
Yes, yes, I did mention that this was
But
the case.
yeah, I understood. But you mentioned there ki it was having like off target
Yes,
effect.
I mentioned.
If you have given a suitable plausible explanation, then it should not be a problem. Shouldn't
It's not just from the like not just the assignment
Yeah,
point of view.
I understood your point. In these cases, we look for different tools and maybe other, even in worst case scenario, we design it manually.
Okay.
है ना? Okay.
Sir, hello.
Hi, Soumya.
Hi, sir. So, in the slides, in at many places, many PDFs or many links were mentioned of Thermo Fishers, for example. So, it was mentioned we should refer to that, but it contained a lot of data. So, for exam point of view, we can definitely refer to it, but do we need to learn that much of data given or
For
any
example,
protocol?
which one you are talking about?
So, just a second. So here an Agilent PDF was given. It was gene morph to random mutagenesis kit ka kuch instruction material. There is something
Okay,
like that.
Asta is saying that is just for reference. You don't need to pay too much heed to that. Just for
Okay,
reference, okay?
okay. And similarly, the lipofectamine one, the protocols and clubbing.
Lipofectamine, I think you should look at it, the official document, Thermo
Okay,
Fisher
sir.
ka, whatever
Yeah.
they're suggesting in different, like how to scale up, how to scale down, how much
Okay.
microliter of lipofectamine you should use per surface area or number of cells. That thing, I think it is important, but then again, you don't have to like go into too much depth. Like whatever Rahul covered, like in during his lecture, that should be fine, I think.
Okay,
Okay.
sir. And yeah, so whatever was covered in the lecture, that should be fine. I mean, and we don't need to. I mean, we'll definitely study for our majors and all that.
Right.
So,
Right.
but lab slides would be enough for the exam point of view, I suppose.
Yeah, your practical exam should be covered by that.
Okay,
Okay,
sir.
but
Thank
your
you.
major, we don't have any role in your major. That is completely
Yes,
up to them, right?
yes,
Okay.
yes, sir. Thank you.
Okay. Anybody else? Okay, one more time. Anyone?
So one more question.
Yeah, sure,
Uh,
sure.
just that in the GFP transfection in cell, it was said that we can do stable as versus transient transfection. So, is there any definite? I mean, does it happen randomly that in some cases stable transfection is
No,
seen,
no,
in
no,
some
no,
cases?
absolutely not. See, whatever transfection it usually done, or I think taught to you is the transient transfection, whose effect lasts for at most 7 days. After that, the effect
Okay,
is
sir.
not there because after each cell division, the amount of plasmid being replicated to the cells, daughter cells, is becomes less. Okay, so gradually the cell plasmid is lost, and whatever your effect was there, it will get lost. But the its lingering effect will be there, like some pathway was switched on and it will be continued to be on. But the thing is, for stable cell line preparation, what you need to do is you need to have a plasmid in which your gene of interest is cloned, and this plasmid has a, for example, a marker, for example, puromycin resistance marker. Okay, you need to linearize that plasmid and transfect it into the cells. So in that cases, what happens is sometimes some copies of the your gene of interest will get integrated into the chromosome, and then you give those cells like a selection pressure, like you treat them with puromycin and you keep culturing them. So after some few passages, only those cells will be left which have stably integrated that particular gene of interest. So that is how you make a stable cell line, and it is not by chance ki, some cells will become stably transfected and some will be transiently transfected. That is not, absolutely not by chance. You have to take good care, and it's not easy to make a stable cell line.
Okay, sir. Thank you.
Okay. Anyone else?
हेलो सर। सर कितने क्वेश्चन पूछे जाएंगे?
अच्छा, शिवम किस में?
माय प्रैक्टिकल क्विज में
कितने मार्क्स का होता है वैसे?
25।
25. I think maybe 25 questions would be there, but it is completely ma'am's wish. She can make like two, three questions of you, 1 marks or 2 marks. It's up to her completely. We yet don't know yet.
Okay, sir. Thank you, sir.
Okay.
So will there be negative marking as well?
Yes.
Oh, it usually there is there is.
Usually there
Okay,
will be.
okay. So expect negative marking.
All right, sir.
Okay. When is your exam, by the way?
9th
8th,
November. 9th, I think.
8th, I think.
Ninth
I,
morning.
9th.
And is there any deadline of your assignment, like marks?
No.
Okay, cool. Anything else? Don't worry, you will do good in practicals. I hope. Okay, if that's it, we will close the meeting. And there is a Google Doc shared with you guys. Even if some of you couldn't join, but you have a doubt, you put it there. Uh, we'll try to get back to you. This link will be open up to your exams. Okay.
Thank you.
thank you, sir.
Thanks a
Okay.
lot.
Bye bye. Bye
Bye,
bye.
sir.